The citric acid cycle is the process that comes after glycolysis and pyruvate processing. The, the purpose of the citric acid cycle is to produce FADH2 and NADH, which will be used by the electron transport chain to create more ATP, thus having more energy for the cell. The intermediates of the citric acid cycle can also be used to generate other types of molecules, such as nucleic acids, proteins, and etc. Another key thing to note about the citric acid cycle is that it is a cycle, and anything that will be used will be regenerated and nothing will be lost. So we start off with pyruvate, and pyruvate is a product of glycolysis. So remember, we start off with that sugar molecule that is oxidized into two pyruvate molecules. Now, one of those pyruvate molecules we will focus on will be converted into acetyl-CoA through pyruvate processing, in which the enzyme pyruvate dehydrogenase will remove a carbon from pyruvate, and pyruvate is a 3-carbon molecule and acetyl-CoA is a 2-carbon molecule. So we go from a 3-carbon molecule to a 2-carbon molecule, and we attach a coenzyme A group to the pyruvate, and we now have acetyl-CoA, and this process was catalyzed by pyruvate dehydrogenase. Now, when acetyl-CoA, it can now be used in the citric acid cycle. It combines with oxaloacetate. So oxaloacetate is a 4-carbon molecule and acetyl-CoA is a 2-carbon molecule, and it combines to create a 6-carbon molecule known as citrate. This reaction is catalyzed by citrate synthase, and in this process, that coenzyme A is actually lost. So that coenzyme, so on acetyl-CoA, that coenzyme is lost, and we're left with these 2 carbons that bind to the 4 carbons of oxaloacetate, and now we have citrate. Citrate will be converted into isocitrate through two sub-steps. In the first step, we use the enzyme aconitase and we remove water. So essentially, on our citrate, we're removing an OH group and we're removing a hydrogen to gain water. So we lose the water, and then we get cis-iconictate, and then we reintroduce the water, and the OH and hydrogen will attach to opposite areas. So they flip, so they isomerize. So we went from citrate to isocitrate by first removing the OH and H group, and then reintroducing the OH and H group, and they're in the opposite positions. So we have an isomer of citrate. Now, isocitrate will then be converted into oxalosuccinate through a dehydrogenase enzyme. A dehydrogenase enzyme is commonly used when we are reducing an electron carrier. So here we can see we have NAD plus and we are reducing it to NADH. And we are doing that through the isocitrate dehydrogenase enzyme. So by losing the hydrogen, we are becoming isocitrate to oxalosuccinate. Now, oxalosuccinate is going to undergo another reaction in which we're going to produce alpha-ketoglutarate. And we have the same enzyme, isocitrate dehydrogenase, and we are going to be losing a carbon molecule. So remember that up until this point, we had a 6-carbon molecule, because when oxaloacetate and acetyl-CoA combined, we had a 6-carbon molecule citrate. Isomerization reaction, we still have 6 carbons. We reduce NAD+, we're still 6 carbons, but now we're losing a carbon. So alpha-ketoglutarate is a 5-carbon molecule. So far we've created one NADH molecule. So alpha-ketoglutarate will undergo another reaction in which it's going to produce NADH, so it's going to reduce another NAD+, and it's going to be another dehydrogenase enzyme, but in this case, it's going to be alpha-ketoglutarate dehydrogenase. So once again, we're using a dehydrogenase enzyme, so we're reducing NAD+, and we're also producing carbon dioxide, so we're losing another carbon. So now we're going to be left with a 4-carbon molecule. Succinyl-CoA is a 4-carbon molecule. So we've lost the additional carbons we added. Remember, acetyl-CoA was a 2-carbon molecule, and that was the additional carbon we were adding to this process. Now we've lost those 2 carbons, so now we're back to the original quantity of carbons that are found within the citric acid cycle, because remember, oxaloacetate has 4 carbons. So now succinyl-CoA has 4 carbons. And also what happened in this process where we were reducing NAD plus and producing carbon dioxide via the dehydrogenase enzyme was that we were also attaching a coenzyme A group to succinyl-CoA. So succinyl-CoA, once it's produced, it's going to then create succinate, in which it's going to lose the coenzyme A group. And as that coenzyme A group is released, there is a release of energy. And that energy release is going to phosphorylate the GDP, and it's going to become GTP. And this is done by the enzyme succinyl-CoA synthetase. So that energy release from that coenzyme A group catalyzed the reaction of GDP to GTP. So a phosphate was essentially added. And now we have succinate. Now succinate will be converted into fumarate via succinate dehydrogenase. So we have a dehydrogenase enzyme. So we're going to reduce an electron carrier. In this case, it's going to be FAD. FAD is going to be reduced to FADH2. So now we have fumarate. Now fumarate is going to be converted into malate via the enzyme fumarase, and water is going to be introduced into this reaction. Once we have malate, malate is then going to utilize the enzyme malate dehydrogenase, which is going to produce, once again, a reduced electron carrier. So we have NAD plus, and it's going to be reduced to NADH. And now we end up with our original molecule oxaloacetate that we started with. So in this reaction, we take 2 extra carbons because acetyl-CoA has those 2 extra carbons. We combine it, we get this other molecule, citrate, which is a 6-carbon molecule. When we lose those 2 extra carbons that we gained, we go back to our 4-carbon state, and then we regenerate oxaloacetate. So in the end, nothing is lost. It's a cycle. Whatever we used, we created it again in the end. And in the process, we produced 3 NADH molecules via the dehydrogenase enzymes. We lost 2 carbons via carbon dioxide through dehydrogenase enzymes. We produced 1 FADH2 through a dehydrogenase enzyme. And we also produced 1 GTP molecule. Now the NADH, the 3 NADHs, the 1 FADH2 are going to go on to the electron transport chain to produce ATP, and that GTP can be utilized for energy as well. And that is essentially a brief intro into the citric acid cycle. It's a cycle which produces reduced electron carriers, which will be used by the mitochondria to produce energy through the electron transport chain and chemiosmosis. The citric acid cycle is a process that cannot occur in anaerobic conditions. In anaerobic conditions, we will see fermentation processes such as lactate production and ethanol production. This process occurs after glycolysis and pyruvate processing.